basics of genetics. So this is a very broad topic. As you feel, you can speak about it one semester or even more. So this is very hard topic, but interesting topic. So I really like it. And therefore, I hope that you will find at least something which might be interesting for you. I will go through and step by step the presentation which I have prepared. And we will see. If you have some questions, something is unclear, something is less understandable, just ask. So that's it. So of course the video is recorded and you can see it later on. Also, I have prepared the mini test, which is checking your understanding to the topics of today's lecture. First of all, today we are going to talk about some basic concepts. We have to clearly know what means heterozygote, homozygote, what is variability, heritability, some keywords which are essential for the understanding to genetics. I will just pay attention to the heritability in contrast to variability, of course. Also, I will a little bit touch to the creation of mutation. And also my favorite topic is evolution. So therefore, I just prepare a few slides from the population of genetics of population. So this is something which might be also fancy for you, because this is these theories are considering about the origin of humans. So it might be interesting. I hope so. So first of all, what is the difference between heritability and variability? So heritability and variability are two extremes conditions, actually, or terms. Heritability is determined by our genes. This is what is given. This is what we have from our parents, from our ancestors, and we cannot change it simply. This is quite difficult. So this is something like prescription for us. And we are working with our genes. And this working with our genes give us to space for variability. So it means that there are two pressures. The first one is heritability, as something which is strictly given. And contrary to this, works the force of environment, culture, our lifestyle. So in the end, the result, what we are, how we look like, how healthy we are, how clever we are. This is the result between two extreme features, heritability and the variability. So they are meeting somewhere in the middle, or it depends. Sometimes the genes are too strong that we cannot practically do anything, and we are staying somewhere here. On the other hand, our genes are weaker than the pressure, the pushing of the environment, for example, and therefore we can get with the result more to the variability. So also the pressure of environment, for example, it's not constant to all our genes. So this picture can be drawn for every gene, every feature in our body. Heritability is something which is constant, something which gives to species some continuance, yeah? Some similar features, the shape, for example, hand. The human hand has 5 fingers. There is a thumb which is a little bit different than the rest of the fingers. This is a really conservative feature, very conservative trait. However, if you just see your hand, your colleague hand, the shape is a little bit different. Someone has longer fingers, someone has bigger hands. So this is the variability, which really sometimes depends also on the work which we are doing. There is absolutely different hands from people which are working with hands. They have much bigger hands, much rude hands. In contrast to, for example, scientists or teachers, we have very soft hands, little one, tiny fingers. So it really, this is just the adaptation to the environment, to the work which we are doing. Variability, it stands in contrast to heritability. And this is just the basic assumption for evolution. Without variability, there is no evolution. Every generation, the humans or animals or plants in one species would be the same, but it's not the same. So therefore, we know that variability exists, and this is the driving force for evolution. So it helps to combine new genes and to create new genes, also to combine some genes, creating some new type of a feature. So this is just something which is not really well discovered, but we know that it exists, and this is the driving force for evolution. So what about features? We have several types of features. We can talk about the formative. We can talk, it's in general, what is a feature or trait? Trait or feature, trait. Maybe this is much more common, trait. So this is in general a property of an organism, something which is characteristic for us. It could be a nose, color of hair, structure of hair, color of our eyes, but also the shape of our eyes, also how we are tall, what is the constitution of our muscles, yeah, how big is our heart, how big is our liver. This is all the features which are coming from our genes and also from the environment, lifestyle. This is just the final product of these two entities. Why the features? Okay, maybe that's enough for the beginning about the features. You can see, for example, here the variability within the population. It's pretty big. You can see this is some kids, some children, blue eyes, reddish hair, brown eyes, blonde hair. So all the combinations are possible, actually. Of course, some features or some genes are dominant, some are recessive, and we'll talk about it later a little bit. So first of all, just to we have to divide features into main parts. The first one is quantitative features. We can measure them. You can collect several people from our city and just weight them or just measure how they are tall. And this is a number. We can put these numbers into a table and say, okay, 50% of Prague population is tall 180 centimeters. 25% of them is tall 170, or it's not 170. We can, the scale is different. Yeah, of course, it depends how we are going to scale, how we are tall. And the scaling is always possible to express as a Gaussian curve. The Gaussian curve could be symmetrical, like this one in the picture. We are talking about the symmetrical Gaussian curve or about the curve with normal distribution. However, sometimes some features, some traits are a little bit different, and they do not follow the normal distribution. So it could be shifted somehow or shifted like this. So we are talking about some skewness and kurtosis of this parameter, which is shifting the distribution from the normal and or Gaussian distribution. So for example, the majority of people has the average feature, the majority of them. Some of them has a feature which is bigger than normal. So for example, the average high from, I'm going to talk about the Czech man, Chickman. So the average is 180 centimeters. So this is this part. So it could be a range from, for example, 175 to 185 centimeters. This is the average of the population. Some little bit smaller part of the population will be a little bit taller, so they will be tall in the distance between 186 to 180, for example. Okay, and the same part will be a little bit smaller. I will use a different color, sorry. Okay, green. Okay. So this part will be, for example, this is just an example. So it will be 170 to 174, for example. And then we can continue on both sides to the smaller people and to the higher people. Yeah. So this is Gaussian distribution of measurable or quantitative features. Is it clear for you?
Yes.
Okay. Sometimes these measurable features can be transformed to some so-called percentiles pictures or percentile graphs. And it's usually used for growing of children. Yeah. So it's differentiated into percentiles. Percentiles means some defined amount of percentage, like 95, 85, and something like that, which express the majority of the population. For example, 95% of the population normally ranges in this range. Yeah. The second group of features are qualitative features, quality. We cannot measure it. Here is an example. So this is a nose. So this is different shapes of nose. I know that the picture is a little bit small, but just imagine that everyone has practically different shape and size of the nose. But what is also sometimes it could be interesting that also the shape of feet is something which is quite different. We don't have the same shape of our feet. This is just the selection of the feet which can be observed in European population. So here you can see that the origin of these shapes is Egyptian, Roman, Greek, German, and Celtic shape of the feet. And this is also something which cannot be measured. It's quite rare that you have one of these shapes like Egyptian or Celtic. Normally we have something in between, yeah? Something between Egyptian and Celtic, or between Greek and German. So this is something which ranges from one extreme shape to the second extreme shape. This is typical for all qualitative features. Is it clear what is the difference between the quality and quantity?
Yes.
Okay. So what about genotype and phenotype? Simply, we can say that genotype is something what we have in our genetic information. This is the information inherited from father and mother, and we have this information in our cells. This is given. However, phenotype, this is the result of the pushing of our genotype, of the transcription, and also the effect of our environment. So it's something which was created based on the transcription and also then environment, culture, lifestyle, and so on. Maybe you can imagine that the genotype is the recipe. Just try to imagine a situation. You receive a recipe from your granny to prepare perfect, tasty, excellent Christmas cook. There are some ingredients written in the recipe, and also there is written how you have to prepare it. However, once you realize that in the recipe there is written some ingredients which you are not able to gain, for example in Bila, because some of these ingredients are a little bit different. This is not exactly these ingredients which was used by your granny, so therefore you use some different ingredients. You will substitute the original one, and therefore you have tried to follow the procedure as much as possible, but sometimes you made some mistakes. For example, there is written that the cook has to be cooked 30 minutes, but you just forgot and you are going to bake it 35 minutes. So it means that the result will be a little bit different than the original cake from your granny. So this is the difference between the phenotype and genotype. So genotype is the recipe for the Christmas cake of your granny, and the phenotype is the result you are able to cook from the recipe. So here as an example, so you can see genotype, first is the one in bottom. You can see that, for example, for blue and brown eyes, you need always we have two genes or two alleles for the color of eyes. One of them is dominant. It's in this case the B big, so this one is dominant for the brown eyes, and the B small, this one is a recessive allele. So it means that if we put together B like brown, so it's quite strange in this case that blue and brown it's B, but maybe we can use their different letters, but you know it's brown B, big one, dominant one, and also blue, small b. This is always recessive allele for blue eyes. So this is the prescription. This is the genotype. So for example, you have inherited from your mother and father B dominant for brown eyes, from your father B recessive for blue eyes. However, if you put them together, they are just interact together, and because B for brown eyes is a dominant, it means that it overlaps the effect, the impact of the allele for the blue eyes, and therefore you will have brown eyes. Only you may have a blue eyes if you inherit from your both parents the allele for blue eyes, which is recessive. So this is just the example for the Punnett square. Maybe we can clarify it also in one another example. So just give me a second. I will. Okay. Okay. Let's do it like this. So for example, red blood groups, red, typing. Just think about, I have one question to you. Do you know what is your blood group?
Yes.
Yes, and are you able to tell it to me, for example?
It's A positive.
A
A
positive.
plus.
A plus. So now we are not going to deal with. It doesn't matter if it is A or if it is plus or minus. So just we are going to talk about if it is A, B, 0 or AB. So blood groups. So in general, we have A, B, AB, and 0. This is a phenotype. 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 Okay. However, what is behind this phenotype? This is what we have. What can be measured in a laboratory. But for example, try to imagine that some theoretical example. I am going to draw the Punnett square. Punnett square. Oh, like this square. So everyone has two parents. So father, mother, it doesn't matter which one. So for example, our father has a blood group AB. So it means that we can be sure that he has two genes, two alleles for A and B. Both of them are dominant. However, our mother has blood group 0. 0, it's recessive, so it means that our mother should be or must be also homozygote, having two same alleles for blood group 0. And because of the creation of gametes, so it means that normally the somatic cell has two sets of chromosomes. However, during meiosis, we are going to talk about meiosis next time after Christmas. During meiosis, sperms and eggs are producing the haploid cells. So there is only one set of chromosomes. So there is a possibility that A will be here and here, and the gametes which will be created from your father, the sperms, will be A or B, two groups of sperms. Your mother can produce eggs only with allele having 0. So from these parents, the children will be all of them will be heterozygotes. So it means that they have two different alleles. So allele for 0 and allele for B or allele for A. And because of the combination and contrast between recessivity and dominance of every allele, we can say that this combination will be the phenotype B. The same. This one will be the phenotype A. No another option. Yeah. Is it clear? If not, just ask me. I can explain it again.
Yeah, it's clear.
Also, two girls. Katarina, is it clear?
Yes.
Okay, perfectly. Okay. However, our genes or our DNA, it's not only the genes. Genes are the structures of DNA which serves for the production of proteins. The part of DNA which serves for this purpose, it's pretty tiny, much bigger part of DNA. We don't know the purpose, actually. The majority of our DNA, we are talking about some wastelands of DNA, but this is not good words for these parts of DNA. The reason why we call wastelands of DNA is that we don't know for which purpose we have this DNA. So it's really not fair because it's something which is unknown and it will be discovered later for sure. Now we are finding that there are also some regulation parts of DNA which serves for the production of tRNA, ribosomal RNA, for example, not only for messenger RNA, which serves for the production of proteins, but also there are some regulation parts of our genome which serves for the synthesis of microRNA, which has very big regulatory function in the proteosynthesis, for example. It may serve as a hormone as well as a signalization for some another cells. So it's a big orchestra for the regulation of several processes in our body. And still we know just a little part of this symphony, really. Also, I use several new words maybe for you. I use a gene and allele. So what is the difference between a gene and allele? So for example, if you would like to produce hemoglobin or insulin, some protein, we need some prescription. The prescription is the messenger RNA, which is synthesized from the sequence present in DNA called gene. All genes are present in our genome device. One coming from father, one coming from mother. And because of these two genes may be in general different, you inherited different gene from your mother, different gene from your father, and therefore we call that we have different alleles of one gene. Yeah. It's the same part of DNA, so it means that it's the same recipe how to cook hemoglobin. However, the gene coming from your father could be slightly different than the recipe coming from your mother. Therefore, everyone in our genome has two copies of genes. They might be same. So in the case that the alleles are the same one, genes are the same one, we are talking about the homozygote. But if they are different, so the sequence is different, we are talking about the heterozygote. So gene means the recipe, allele, it's different, slightly different or modified recipes. Within the population, we may have several alleles, yeah, because we are different, we have different parents, and also some populations are very old, for example, the African population. So there are bigger variability in alleles. On the other hand, the European population, we are genetically quite similar. So the variability within European population is much smaller than the variability in African population, for example. Also, locus, is it here, this name? No. Locus is the place where the gene is sitting, yeah, locus. This is the same place all the time in all entities in every species. So this is the place where every gene is sitting. This is really conserved. So we can just conclude this part. So, for example, what is the difference between a feature and a gene? So gene is the recipe which we have. Then we are trying to cook it based on the recipe, and we are using the ingredients which are present in the environment. It's not only the environment, it's also the lifestyle. It's also the culture and so on. And the result is the final feature, how we looks like. how one gene is cooked. Its feature, it might be a protein. I have to mention, because we have two possibilities, we have two recipes usually from one gene. We have two alleles from our parents. So we have two possibilities how to cook one typical protein, for example, protein from insulin. So how we choose that we are going to use the recipe from the mother or from the father? Any ideas? What is the how our body is decided which recipe is used for the cooking of a protein? Which gene we are using, which allele? Any ideas?
Doesn't it depend on like which one is dominant than the other?
Yes, this is the first one. Okay. And if they are the majority of alleles, they are not really strictly dominant or strictly recessive. Sometimes it's a cooperation among them or between them. And what is the driving force? What is the most important in this case, that they are not strictly dominant or strictly recessive?
And the environment.
Yes, definitely. Maybe you know something about epigenetics. Epigenetics or DNA methylation. DNA methylation. So this is very important. DNA methylation. So methylation, it's some chemical process. So it's just the addition of methyl group to our DNA to block transcription of one allele. So it means that we have, once we are born as a child, as a newborn, we have practically no methylation in our DNA. So it means that both alleles have the same chance to be used as a recipe for a protein. However, within the time, how we are growing, we are getting older and older, the number of methylation is increasing. So by the level of DNA methylation, we can assess quite precisely the biological age of us. And the main point of methylation is to simplify the protein synthesis process. So it means that one of the allele is more usable, there are more advantage to synthesize exactly the protein according to one of the allele. Yeah. So that's the reason that the second allele is blocked by methylation. Is it clear?
Yes.
Yes.
Okay, great. Molecular genetics. This is a pretty new, well, it's not new, but very, very dynamic scientific field. And this science really focuses on the basics of genetics, but on a molecular level. If we just talk about the Mendel genetics or some of the older or traditional genetics, we are talking about features, about the pea color of blooms and flowers and so on, about the shape of seeds. But molecular genetics is more focused on the genes, on the sequence in our DNA. You can see that there is the sequence of nucleotides. This is what is interesting for molecular genetics. Just try to understand that really DNA serves as a cookbook for the proteins which are made us. However, the informational capacity of our DNA is much bigger than we need. So we are using something like 10% of our DNA. And that's quite amazing. We practically don't know much about the genetics, because if we know or recognize only 10% of DNA which is placed in every our cell, this is something which is unknown, and we have to really discover it more. So if we put together all the genetic information of our living organisms in our Earth, so it has just the pinhead size, pinhead size, it's pretty tiny. And this is the information-rich substance which contains many information. You cannot imagine how rich this substance is. So, what about karyotype and locus? I already mentioned that the locus is the place where the gene, so in this case alleles, 2 alleles, are sitting. This is conserved. We have within a species, we have quite conserved karyotype. For example, for humans, we have 46 chromosomes, so 2 sets of 23 chromosomes. Yeah, this is the sex chromosome 23. However, in our planet, in the Earth, we have some species which doesn't have really the same number of chromosomes. Why? If we have the stable number of chromosomes, it means that the species is already just behind its evolution. Yeah? It's really evolutionarily stable species. But recently, for example, in Madagascar, there are living some monkeys, which are called lemurs. Within the species of lemurs, there are a variety of species of these monkeys. However, the number of chromosomes, it's not the same one. It differentiates from one generation to another one. So it means that lemurs, such a big group of monkeys, they now provide so-called genetical drift or genetical drifting. So it means that this species is extensively developing. For example, if we get back to humans, if we compared the number of chromosomes of chimpanzees, our closer relatives in animal part, they have 48 chromosomes. We have 1 pair less, so we have 46. So we expect that our ancestors, which were together of chimpanzees as human, had 48 chromosomes, and within the evolution we lost 1 pair of chromosomes. So this is expected, but it happened in really past. So now this is the topic which I would like to a little bit touch only. The first reason is that you just saw it in the video, the segregation and combination of chromosomes. And the second reason is that we have a new student which didn't participate in the semester, and I expect that she would like to receive the credit, and therefore she would like to prepare the presentation in January a little bit longer. So I think it's a good topic for her just to focus on the traditional genetics, and to the traditional genetics segregation really belongs. So this is something which I will leave now, but it is in the video nevertheless, and also we can return to that after Christmas. So again, combination segregation. Now we are getting to the question of autosomes and heterosomes. So I have mentioned that we have 23 pairs of chromosomes. 22 of them are autosomes. So it means that they doesn't depend on the sexual difference. The last one number 23, is different. For women, it's X. For men, it's Y. So it's very important, the sexual determination based on this gonosomal chromosome. This is the first point. However, some diseases may be inherited only depending on the distribution of the X or Y chromosome. So during meiosis of men, so the production of sperms, you are producing 50% approximately of sperms having sexual chromosome X. This one is coming from your mother. And the second half is Y chromosome. So you have, okay, it's Y, 50%. So there's 1-to-1 chance to have boys or girls. Yeah, so you have girl sperms and boy sperms. Actually, you have slightly bigger amount of Y sperms because this is the reason is that nature, there's a problem that the Y sperms, they are more sensitive to the changes of the environment. So therefore you are producing a little bit more, 52 for example, and here is 48. But it doesn't matter, this is just the difference. It's quite interesting that older men, they are producing more X sperms than the Y sperms. This is also very interesting, and also men living in very bad environment, very toxic environment, they are also producing more X sperms. So meiosis of women, women. We cannot produce Y chromosomes or X with Y chromosomes, so we are producing 100% of X chromosomes. Yeah, 100%. So it really depends which sperm is the first one. If we are asking for a gender or if we are considered with the gender. But there is much more important things. Some of the features, some proteins are encoded only in the Y chromosome, only there, only here. The Y chromosome, in the X chromosome, sorry, in the X chromosome, only in the women chromosome. The X chromosome is much bigger than the Y chromosome. So the amount of genes placed in Y chromosome is much smaller than the amount of genes present in X chromosome. So it can happen that, for example, the mother, which is producing the X with X chromosome, has one gene there which has some mutation, or some mutation, for example. Yes, there is some problem with the gene, so the gene is invalid, for example. However, if the sperm has only the Y chromosome and get in combination with the X coming from the mother, where is the wrong chromosome, the invalid one, it means that the boy which is born would have unfunctional gene for one protein. And sometimes it might be related with some pathological changes. Okay. For example, hemophilia. This is one example. That the mother or women, that they are carriers. So she has one wrong X chromosome, so she can produce healthy eggs, 50% of healthy eggs, but 50% of unhealthy eggs. If the unhealthy eggs just meet healthy X chromosome from the father, there is no problem. The daughter is fine. She is also again the carrier. But what is a problem if this unhealthy gene meets the Y chromosome? So if it is a boy, the boy is affected. This is just the example. For example, in past 100 years ago, we had the family from Russia, Caesar Nicolais, and his son. His son was affected with hemophilia. So they had only one son, I think something like 4 daughters, but one son, and this son was affected because of the presence of the wrong gene coming from the mother, which hasn't no alternative in the Y chromosome. So the biggest problem is that in Y chromosome, sometimes it's not the alternative to the genes present in X chromosome. Clear? Questions? Okay, no questions. This is just some basics. Here you have Johann Gregor Mendel, which was the scientist working in Brno in Moravia in the Czech Republic, and also Hugo Maria de Vries. Both these men, they discovered pretty much about the genetics of organism, but they focus on the features. They didn't know what is behind. They expected some genes, of course, but they haven't the apparatus of the molecular biology or molecular genetics, which can help them to understand on the molecular level what happened during heredity. But it doesn't matter. The apparatus they have provided, it's pretty amazing, pretty interesting. They are the fathers of genetics. I think, heredity of qualitative and quantitative features. So quality and quantity, we are a little bit getting back to the first slides, which I mentioned. So quantity, it's more or less better understood than the quality. So the quantity, how we are tall, this is something which has very good support from our heredity. So it means that how we are tall is inherited from 90% based on our parents. So there is a little bit effect of environment, but however the effect is only 10%. Much bigger effect of environment can be observed in the heredity of qualitative features. So it means the shape of hands, the shape of nose, and so on. How the feature is created. Usually it's not just one gene and one feature. Usually the feature is provided by more genes together, by the cooperation of more than one genes, usually. Also, I already mentioned what is homozygote and heterozygote, so this is important, just to remind you that homozygote contains the same alleles of one gene, the same one from the father and mother, and heterozygote contains a different one different from father, different from mother. About dominance, so sometimes we just from grammar school or from high school we expect that there is absolute dominance or absolute recessivity. For the majority of genes, this is just the presumption. However, it doesn't work for the majority of genes. It's working for the blood groups, for example, but it is less valid, for example, for the color of eyes, color of hair, and so on. We are talking about so-called partial dominance. So it means that one allele is slightly less dominant than the other one. And the final feature, the final trait, is created by the cooperation of these two genes, these two alleles. Also, we can talk about so-called intermediality. So it means that there is no dominance, no recessivity. The effect, the impact of both alleles is 50-50. Autosomal heredity. We are talking about sexual heredity and autosomal or gonosomal heredity and autosomal heredity. Autosomal heredity is focused on the heredity or the transfer of genes which are placed in somatic chromosomes, so 22 chromosomes, 22. And this heredity is not fixed to the gender. Yeah? This is important. Gonosomal heredity, we are talking about only the about the last chromosome, so about one. Its number is 23. We can distinguish two heterochromosomes, X or Y, and that's the problem sometimes, which I mentioned, with some diseases which can be inherited and just observed for the sons or in the man population. Okay, what about the quantity? So it's something which can be measured, and just to remember that the distribution of the genotype is given by the Gaussian curve, and there is also very high effect of the environment. However, there is one exception. This is the man height, how we are tall. So this is the effect is of the environment is smaller. But for example, our weight, it really depends on the environment and more on our lifestyle. So just be careful with this categorization. But just remember that quantity, it's a number, and we can draw for the quantity or quantitative features a Gaussian curve, which could be asymmetrical or could be shifted to one or the second extreme. Is it also possible? But the qualitative feature, it cannot be expressed just with one number. Yeah, it's just a question of shape, structure, and so on. Again, we are getting to the heritability. There is a coefficient which can be called heritability coefficient or variability coefficient. Its ranges from 0 for 1, so just heritability is opposite to variability. So for example, if heritability is 1, variability is 0. If heritability is 0.8, variability is 0.2. Yeah. So if they are counted together, it's always 1. So they are ranging from 0 to 1. If the heritability coefficient is approaching to 1, we are talking about a strong heritability. If the coefficient, heritability coefficient, is approaching to 0, we are talking about low heritability coefficient. So it's just the fight between genome and environment. This is what I already mentioned, the variability. It's the main driving force of variability. It's environment, lifestyle, and culture. However, there are also some external effects which are inducing, for example, mutation. Mutations are random, but there is also crossing over. This is something which nature is getting the inspiration or creates a new variance, and this is just a stochastic process. mutations as well crossing over. But it's not 100% true that crossing over is a stochastic process. Now we know, we expected that maybe 20 years ago. But now we know that there are some groups of genes which are herited together. So it means that we have a chromosome, for example. I will just draw a part of it. And we have here some genes. Okay, I will draw the genes like with a line. So they are genes, and different color I will need. Okay, so these lines are genes. However, these red genes, which are close together, they are herited together. So it means that the crossing over is not really the stochastic process. During crossing over, we can observe that this part of the chromosome is not breaking and change with the second chromosome coming from the mother, for example. Just in any point of DNA, it means that the whole part which contains, in this simple example, 3 chromosomes is just breaking and crossing over together. Yeah, this is not valid for all chromosomes or for all genes, but it could be. It depends really. Sometimes we may observe, for example, that some features are grouping together. For example, if our grandfather has curly hair, black hair, and brown eyes, and the genes which are really encoding these features, they are together, it's a very big chance that these genes will be herited together. Yes, sometimes also, if it's not necessary that the genes are only about our appearance, it could be also our talents, our disposition to provide mathematics, music, and so on. So crossing over, it's not 100% stochastic. There are some regions of genes or sets of genes which are herited as a package. What about mutations? Mutations are really stochastics. There is no really rules or something which could just control them and order it. We can distinguish the mutations which are in points. So it means that there is a changes in the sequence of nucleotides. So it means that one nucleotide is just substituted with another one. This is the first example. So there are chromosomal mutations. There are some changes in chromosomes. There some genes, one or more genes, can be lost or duplicated, triplicated, and so on. So it's about the translocation of genes between chromosomes and also about chromosomal aberrations. The biggest changes or biggest mutations are in the range of whole genome. It affects usually the number of chromosomes. There are 2 variants, aneuploidy, so it's a loss of one chromosome, or ploidoploidy, it's just gaining one additional chromosome. This is the example of polyploidism. So it's Down syndrome, which is caused by the triplication of 21st chromosome. So normally we have 2 copies of 21st chromosome, we have 2 copies of every chromosome, but people suffering with Down syndrome, they have 3 chromosomes from the 21st chromosome. Okay, how the mutations are caused? There are several reasons. Spontaneous mutations, they are random. This is just the mistakes during the replication on transcription. Sometimes there is an effect of mutagenous factors. However, mutations are quite rare because the we have very strong, very functional, very effective repairing system or checking system, which is checking if the DNA was replicated correctly. There is a problem in the case that if there is some induced mutation, the mutation can be induced by some radiation, by some chemical agents, and also by humans. So probably you know some CRISPs. This is actually also some technique which allows to change the DNA sequence. But usually the CRISPs techniques, they are used for some good purposes, hopefully. Now we are getting to my favorite topic, to the population genetics. Based on that one, we may distinguish two types of population, panmictic and inbreeding population. The panmictic population is the population with sufficiently wide population where the random crossbreeding is allowed. So it's, for example, the Czech Republic population, European population. There is no restriction. Everyone can be married with anybody and have with them children. From the genetic point of view, this population is not changing. There is some steady state which is reached by this free or random crossbreeding. On the other hand, there is the inbreeding population. Inbreeding population is characteristic by low number of entities, low number of animals, humans, monkeys, elephants, which cannot provide the random crossbreeding. So it means, for example, that the number of the entities of humans is too small. For example, the group of 30 people, 15 men, 15 women, this is pretty small group. And within the time, there is a big risk that the close relatives will be inbreeded, will be married, and will have some children, which might be affected somehow. So this is typical example of this inbreeding population is European nobility. Probably you know that if the Czech kings, the British kings, Dutch kings, Spanish kings, all of them, they are relatives together, because it really depends in past on your social status. You cannot marry everyone. There are some groups of people which were higher positioned, and therefore they provide the marriage only among this quite small and limited group of people. The risk of inbreeding is just that the close relationships and breeding in close, like incest, something like that, would provide some badly affected entities, badly affected children. However, there is another aspect of this inbreeding. We expect that in the past, it has a big effect on our human population. We expect that in quite long past exists a group of humans which counted something like 50 or 60 people together. They were isolated somewhere, and therefore they provide this inbreeding. However, this inbreeding has a positive effect to the extension of our brain. So we are talking about the bottleneck effect. Bottleneck effect. Effect. Okay, bottleneck effect. So not all the time the inbreeding has the negative result. Sometimes it might, by some big portion of fortune, it can finish with some success, or with just finish by some good result. That's it. So how we can just disrupt the genetic stability. So the panmictic population, this is quite stable within the time. So there's a there is reached some steady state. However, also the steady state of the panmictic population can be disrupted by some effects like mutations, but also selection. So just think about it, how we select our partner. So it really depends on our sexual preferences, and also the selection. It depends how we are adapted to the conditions, environmental conditions. Also the genetical drift, which is usually related with migrations of populations. And now I would like to just show you a few books which are very important and very interesting. This is popular books. It's not the scientific articles. They are too complicated for you, but these books, they are written for women in like housewives, something like that. So it's easy to understand them. For sure, I can recommend you the books from Charles Darwin, Natural and Sexual Selection. Also the Red Queen from Matt Ridley, which consider the evolution of humans. And a very good book is from Jaroslav Lager, Frozen Evolution. So it means, for example, Charles Darwin just consider the evolution like a smooth process. However, Frozen Evolution expect that if we are not pushed by the environment or some circumstances to be changed, we are remaining all the time the same one. Then something happen, the selection is stronger, therefore we are changed. So therefore the evolution is frozen in these linear parts. Then by a jump something happen, and then we again continue with just to do anything. Also very important or very interesting is the genealogy, which is based on the Y chromosome, and also in the mitochondrial DNA. Also genealogy according to the languages, and of course the theory out of Africa. So migration of Homo. There are several approaches how the scientists consider that we get from Africa, but probably the first Homo sapiens, it means that Homo habilis, Homo erectus, and Homo sapiens. These three Homo were created in Africa, all of them. We expect that Homo habilis didn't migrate, just remain in Africa. Homo erectus was spread from Africa to the rest of the world. However, there were a second wave which was provided by Homo sapiens. These Homo sapiens developed probably by the bottleneck effect somewhere in Kenya, in the Lake Turkana. And from this part, they spread and colonized the whole world. Also, there are some interactions of between these Homo subspecies also again, because Homo subspecies, for example, Homo sapiens sapiens, I will write it Homo sapiens sapiens, as we are, and the second one, his cousin, was Homo sapiens neanderthalensis. It is considered that Homo sapiens neanderthalensis get first from Africa and colonized only Europe. And the second step or second wave, Homo sapiens sapiens, he recolonized Europe and also rest of the world. How we know that? Maybe I have to speed you up. Oh, just 5 minutes, okay. I will. So we are talking about the Stone Age, about Paleolite, Mesolite, Neolite, and the creation of Homo sapiens. We expect that the Homo sapiens was created 100,000 years in Africa. So that's the valid theory, and this is supported by many sources of information, archaeology, also genealogy, also molecular genetics. What about mitochondrial DNA? You know that we have our chromosomes, our DNA, which is placed in nucleus, and also we have some DNA which is not placed in nucleus. This is the external DNA which is placed in our mitochondria. This mitochondria is not the eukaryotic DNA, it's the prokaryotic DNA, it's circle. And we inherit this mitochondrial DNA only from mothers. How it is possible? The egg contains mitochondria, also nucleus, and other organelles which are usually present in every cell. However, spermia, it has genetic information, DNA, nuclear DNA, and here is the neck, which is rich on mitochondria. However, once spermia reach egg, and we know what happened later, only the genetic part is getting to the egg, just phagocytated by egg, and the chromosomes are splitted, crossing over, and it continues as we know. The rest of the spermia remain outside the egg, and also the rest of the spermia, they are the mitochondria. That's the reason that 99.9% of mitochondria are coming from mothers only. This is good information because we can use this circular DNA for the genetic mapping of our relatives. For sure, for the mother line, fathers are standing a little bit behind, but don't be sad, Y chromosome can be used for the same purpose. They did some studies because it was scientists, they are curious, they are like small kids, they are playing. They were asking if we have some DNA coming from Neanderthals. Based on the analysis of mitochondrial DNA, it was proved that we have no DNA coming from Neanderthals. However, if they provide the same analysis using the Y chromosome, they found that our DNA contains something around 2.5% of DNA. This DNA, this little amount, 2.5%, it's not pretty much. If we consider that, for example, 8% of DNA which we have in our nucleus is coming from viruses, is responsible for our higher effective immune system, for example, but on the other hand, for some higher probability to suffer with cancer, for example. So, what about the roots, genetic roots of our of inhabitants of Europe? Are we farmers or hunter-gatherers? Scientists made this research and they found that for 80%, more than 80% of our mitochondrial DNA is coming from the hunter-gatherers. Only between 15 to 20% of mitochondrial DNA is coming from Middle East and probably get to Europe with agriculturing from the Middle East. Also, this theory is confirmed by the analysis of Y chromosome. Also, from the comparing of the European populations, it was found that Basque, you know, it's the inhabitants living in Pyrenees, something is the region between France and Spain. They are a little bit different than the rest of Europeans. For example, the biggest difference is Rh factor. Normally in the world population, 85% are RH plus. However, in Basque, for Basque, and okay, 15% is RH minus. However, they have different incidence of RH minus. RH minus for Basque is 80%. This is the practically opposite one than the rest of the Urums. Also, the language is not similar with the Indo-European languages. And the DNA, the mitochondrial DNA of whole Europeans contains also the DNA from the Middle East. However, the Basquian DNA, mitochondrial DNA, doesn't contain this DNA coming from the Middle East. So we expect, based on this theory, that the Basquians, they were the original Europeans. And now the last thing which I would like to mention is the theory of 7 daughters of Eve. So this is the theory which was popularized by Brian Sykes. He found, based on the molecular analysis of mitochondrial DNA, seven epicenters of or 7 women which were in the beginning of whole European population. For example, here in Pyrenees, Helena, which lived based on the calculation contrast between the genetic clocks and the normal clocks, they found that she lived in Europe 20,000 years ago. 42 or 41% of whole European population carries the mitochondrial DNA coming from this lady. Only these all women, they are Xenia, not Xenia is belonging here. Only Yasmin, she came to Europe as the last one, and probably she just bring a new knowledge how to plant, how to provide some farming agriculture. So here you can see Helena. She has the biggest population within the Europe. So, questions? Okay, if not, I will.
May I ask?
Sure.
Is because you mentioned something about like genetically, you know, having some skills genetic. Could IQ be genetic? Like if you're born to high IQ people, are you likely to have higher IQ as well?
Yes, IQ is quite artificial parameter, but something like intelligence and IQ, yeah, which is characterized by IQ. You know, IQ, it's a really artificial parameter which was provided regarding to the European or North America population. Some natives like Aborigines and so on, they are not really counting to this artificial procedure. So therefore, it's better to talk about intelligence, and intelligence is heredited really, really much. So, however, if we would like to really develop intelligence, we need also some support from our society, from culture, from environment. So it's not enough to be clever only, to be intelligent. We have to develop our intelligence. Is it clear?
Yes, thank you.
Good, but good question. So I am going to send you the mini test. So, okay, chat is here. So, is it okay if I send it here? Could you see it? Or I will put it also.
Send it to the email as well.
Okay, to the email, but a little bit later. I have now discussion with a new student, so you have to wait. Or I will put it to Facebook. Is it okay, Facebook? You know,
Yes,
it's quite long procedure with email, because I have to log to SIS, find you as a student, write an email. So, the biochemistry, biology, we are here. So, but for sure I will send it by email, but maybe in the evening, okay? So here is the questionnaire. I wish you now and I am looking forward to you first week in January. So I think the date is 7th of January. So please prepare your presentation. If your presentation should be 5 minutes, just 4 slides, 5 slides at maximum. Yeah. And I will give you also the first week in January the credits. Yeah. Everything is clear?
Yes, thank you.
You are welcome. So have a rest, enjoy Christmas, and see you after Christmas.
Thank you. Goodbye.
Bye-bye.
Bye.
Thank you.
Goodbye.
Hi, Sam.